I would like to introduce our next speaker. She'll be virtual, but we're excited to have her here. Dr. Paula Leon is a professor of cell biology and neuroscience and director of the Cell and Gene Therapy Center at Rowan Virtua School of Translational Biomedical Engineering and Sciences and Virtua Health College of Medicine and Life Sciences of Rowan University, New Jersey. She is internationally recognized as a pioneer of gene therapy directed to the central nervous system. She conducted the first gene therapy study for a clinical neurological application ever approved by the FDA and funded by the National Institutes of Health. During the past 2 decades, she has built expertise in the clinical application of novel therapeutics to Canavan disease, amyotrophic lateral sclerosis, cerebellar ataxia, and metachromatic leukodystrophy. Her research interests are centered on identifying the pathophysiologic mechanisms underlying neurodegenerative disorders and promoting brain repair. Importantly, her work in gene therapy and white matter disease has led to the metabolic gene therapy strategies being advanced for other neurodegenerative diseases. These efforts have included developing preclinical and clinical applications of novel therapeutics to Canavan disease. So welcome, Dr. Leon.
Thank you so much, Margot. It's just such a pleasure to be here. Thank you, the organizer, and many thanks to Bishan Singh, who invited me here. We had a couple of discussion about drug development and my experience. And so here I am. Thank you so much. So my background is very different because I studied Canavan disease, not ADLD, but the process for drug approval and build-up of natural history is very relative to any drug development for rare disease. And so I thought this would be very much relevant for this discussion. Disclosure, I'm currently a Canavan scientific director and the founder of Myrtle Biotherapeutics, which really started in 2021 and started as a transition between an IND, an investigator IND sponsor transfer to a company that could bring the drug development to the next level, which is all we want. So the discussion today, and I'll go just fast on some topics that you're probably very confident with and comfortable with discussing it. And then I want to really focus on what we're doing now in patients and how we got there. That's the main thing. So within the ASPA gene, there's in Canavan disease, there are inactivating mutations of the ASPA gene, so which result in pathogenic mutations, with an incidence which is 1 in 100,000 in non-Ashkenazi Jewish population, is much lower in Ashkenazi Jewish and within ethnicity that promote marriage within, let's say, the same families. So we have a collaboration going on in Iran right now, for instance, with unique mutations and patients that are affected by the same mutation and keep reappearing. So this is a rare disease, but it's present. There are thousands of patients in the world, all over the world. So what we do know from Canavan disease, the first diagnostic marker is the metabolite that cannot really be metabolized with pathogenic mutation of the ASPA. The sole role of aspartate is to break down the second most abundant amino acid in derivative amino acid in the brain, which is an acetyl aspartate. So the result is elevated NAA in the brain. So a non-invasive test would immediately identify Canavan because the elevation of NAA is very high, and the other test that can be done diagnostically as initial test is the urine analysis. So within the ASPA gene, something that is relevant and important, let's see if this works very well, is that ASPA is solely expressed in oligodendrocytes. However, the byproduct is synthesizing NAA synthesizing neurons. And so there is this, of course, interaction of cell compartments that at one point, especially when the ASPA gene is not, the protein is not functional in catabolizing NAA, myelin doesn't develop, and eventually what we have is really a buildup of NAA and also a disintegration of myelin. The disease starts at birth, and the clinical features are very clear and just a very common, homogeneously spread throughout the population of Canavan patients, with initial hypotonia, head, leg, and microcephaly, seizure, visual abnormalities, and everything that follows, the cascade of events of this of no myelination or no intact myelination in the brain. And then the neuropathology shows disintegrating myelin, brain edema, a severe evacuation. When I discussed it with Bishan, I told him it took a long time to develop what we really wanted to develop. But we started really early. We started with non-viral vectors, which were liposomal vector bound to naked gene, DNA, ASPA DNA, and so we started this early on in early 2000. Then when the technology became available and the viral vectors could be packaged without the use of a wild type adenovirus or with a triple transfection described in the seminal work by Dr. Samosky and Zhao Zhao, then we were able, after 1998, 1999, to even dream to bring AAV into the clinic. And there was a time when in 2001, we received the approval. Finally, the first approval of the FDA for gene therapy for a neurologic disease. And our idea, since Canavan disease is a monogenic disease, only the brain is affected, and primarily white matter. Our target at that time was the reduction of NAA, elevation of NAA. And so we wanted to reduce NAA in the brain, and that was our target. This project was funded by the NIH, was a major effort, obviously, but enable the collection of efficacy data on our target therapy, as well as the collection of natural history data, which is very, very important when we look at therapies. And then later on, and this is starting in 2021, I started in 2018 with the initial interactive discussions with the FDA for a compassionate or expanded access use for 2 patients affected by Canavan disease. The whole program then developed into, we had a in-person meeting, an official meeting at the FDA in 2019 with the families and with the scientists and CMC manufacturer. And the idea was that at that time, the FDA was not prepared to approve this as an expanded access. And so we had to redesign the project and also increase the manufacturing so that we would enable at least 10 patients, or at least what we could with 500 liter production of AAV. So this is where we are, and that's where I would like to bring you, and that's the exciting part. Natural history. This is just one screenshot. We published it, but definitely what we have throughout the brain of Canavan disease, early on, we have brain atrophy. Everything starts with the lack of myelination, to the point that we define Canavan disease as a dysmyelination. So there is no myelin being built normally, not in the typical form of the disease, which really spreads around 99% of the patients are affected by this. Another issue at that time that we published in Science Translational Medicine is that our hypothesis was correct. Overexpressing aspartate using AAV could reduce NAA in the brain. The yellow shaded area represent normal patients, and this is like pretty much the mean of in our patient population, looking at different brain areas. So this told us that since the reduction of NAA did not result in myelination, we needed to target the area where aspartate is really expressed naturally, which is oligodendrocytes. So that took a turn because at that time, even in 2012, there was not a technology that could do that. Very important to say is that during the early phases of these studies, clinical studies, we didn't have an animal model. The animal model well characterized the NER7, which really recapitulate all of the deficit and pathologies that really are displayed in patients, were the one that we share was publicly available. Other mouse models were not publicly available. The tremor rat had additional mutation, is a spontaneous deletion of the ASPA gene and many other genes. So the FDA was not really supportive of that. So we used the most characterized and publicly available animal in all our preclinical data to determine whether a neurotropic versus a targeted white matter just a therapy would in fact make just a difference in terms of efficacy. Of course, toxicology was then done in small and large animal models to fulfill the preclinical package of our IND. So we developed, so the oligovector was developed by Steve Gray at that time at UNC. I was the first person that he called when he identified this oligotropic vector. He knew that there was I was looking for. We collaborated in other projects before. So we clone and optimize HUMA-ASPA gene construct in our plasmid. And the idea was obviously to correct the defect in the compartment, which is normally aspartate expressed. I'm just giving you a screenshot of some of the results that really were published in Molecular Therapy in 2021, showing the comparison between the AV olig, so the targeted white matter therapy, versus the use of AV9, that is a neurotropic vector, and showing primarily that, for instance, we were able to resolve the vacuolation in the brain of the NER7 animal, the Canavan animal, at the lower and higher dose as compared to AV9. In addition, we were able to normalize the development, motor development, looking at really rotarod falls from the rotarod. I heard that Dr. Padiat before was also mentioning this type of studies done in his context, in his animal model. These are very important studies, show efficacy, show justification of proof of concept to move ahead with this technology. So in 2021, the IND was submitted in 2020. There were discussions with the FDA. Finally, the IND was approved December 24th, 2020. And the clinical trial started, the IRB followed almost immediately in 2 months the approval. And the clinical trial started in this beautiful hospital, Dayton Children, where became my second home now. And the project leader there is Dr. Rob Lober. So what we're doing now, and we've now at this point enrolled actually 9 patients, but 10 patients now, because just the patients were treated last week. But 9 patients receive what we call the MIR 101 as the aviolic ASPA, as we publish it as academic investigators. That's what it's called. This is part of a company proprietary name. And I thank Myrtle for allowing me to show you this data today. All patients will follow up for 5 years. Some of the patients and the families, which you see in the demographics, are in fact living in Dayton. Now they moved there, they're happily being cared by everyone in the hospital, doing all of the follow-ups and supportive physical therapy, speech therapy. It's a great hospital for this program. So the injections, based on all the preclinical studies, are delivered ICV, bilateral ICV. The dose is 3.7 10 to the 13 in VG total. This is a human equivalent dose based on the efficacy studies that we have done in humans that proved to be extremely safe in non-human primates. The safety assessment, there are many, of course, and there are efficacy endpoint. Efficacy endpoint that really recapitulate the work that we have done during the past 25 years, where we collected Mullen scale of early learning, gross motor function measure. These are neurologic battery of tests. We have a Canavan neurologic exam. We use a technique that is just very relevant for us. We didn't have it in the previous year, became available in 2021, and this is where we sign up for it, is a synthetic MRI, which really enable volumetric assessment of multiple variables for us, very important: white matter, myelin, CSF, gray matter, and of course brain mass in terms of brain atrophy, as we saw it before, is very relevant. Is one of the marker in Canavan disease that we have in all patients. We also look at NAA assessment, which can be variable, and we're looking at in the brain through NMR spectroscopy and in CSF. And we have a lot of video recordings. We have inclusion and exclusion criteria, of course, as usual. Anything that could be just a cause of additional toxicity or concern, like other medical conditions, antibodies against the AAV, prior intracranial surgery, contraindication to immunosuppression. So these are all, and that's another important point. We decided at this stage in this phase 1, phase 2, to focus on most affected patients. So for the gross motor function measure, patients have to be less than 35% of the total score of GMFM, which means that patients do not crawl, do not walk, have just basically fulfill 1.5 dimension of the GMFM module. And this is just because of risk-benefit ratio. There is an intracranial access, of course, so is kind of is invasive, but we also wanted to make sure that the benefit of the patient, any patient that is below 35%, is a patient that will progress towards vegetative state within years, a couple of years. And so it's important to look at the risk-benefit ratio. While we move ahead, we'll change this, of course, and that's all in discussion with the FDA. Demographic. We have patients that from USA, Italy, Russia, Slovakia, Argentina, Chilean, and we now have United Kingdom, Poland, and other patients that will be enrolled in the next month. This is just looking at the age of therapy and sex. I would like at this point, because the compiled data are just being submitted for publications. And so what I thought I would like to show you one important case. This is a beautiful patient who, through medical visas, and thankfully everything is going well with embassies for renewal and everything else, because with immigration things are changing, as we know. They're still in Dayton, is receiving daily therapy from the hospital. This is all supported by the company. The pretreatment profile is described here to the left. The clinical course, because up to 5 months the patient was normal, that already tell you there is a patient that probably score barely 30 in the GMFM, because normally 99% of the patients are not normal at 5 months. Have no trunk control, have head lag, are floppy. But at 6 months, she starts showing macrocephaly, nystagmus, truncal hypotonia, and then hyperflexia, able to hold the head, but very unstable, able to hold the trunk, very unstable. So it was progressing, like we have seen, and in our natural history, we have many other patients like that. Sorry, I missed something here. Sorry, I'm sure. Sorry about this. Okay. Now, therapy. We just bifrontal trepinations, bilateral. The patients received this with 12 ml of CSF removed from the ventricles prior to the injection. This is an injected dose. The anesthesia is typically 3 hours, and the patients are discharged within 3 days. There were no adverse events associated with the product or the procedure. Now, with PSI-MRI, which we join effort with a team in Sweden and United States, is an FDA-approved test. So any marker of this test is just approved, and is very important because it can be used as a biomarker of disease and disease progression or improvement. In Canavan patients, and we have natural history that is being built now with PSI-MRI. We didn't have it before, but we have conventional MRI showing no myelination in T2 and T1, no improvement in the upper intensity in T2, and just a major evacuation and brain atrophy within the first 12 to 24 months. And in this patient, after the therapy, we saw an increase in myelin, and the increase in myelin is in both white matter and myelin. When we look at white matter and myelin within the context and the compartmentalization and segmentation of the data provided by the PSI-MRI, white matter includes many lipids that can be part of myelin and not part of myelin. So we look at myelin and white matter for both of them, but this is like a huge increase in myelin and white matter. It's just very positive to see it. We have seen it in other patients that have been treated, even patients that were older of this, and soon the data will be published, and so we'll be able to share the good news with the community. Something that is very important for the FDA, what's really important in terms of effectiveness, efficacy of the therapy, and all of the areas of the Mullen scale, in fact, improving these patients, in other patients, as well as the gross motor function. And in my last slide, before the acknowledgement, I'd like to show the patient prior to the gene therapy, and in here, I'll be able to show the patient now that is just walking with some support, doing exceptionally well, and we're thrilled. We're thrilled about this. We've never seen this before. We had another clinical trial done with a neurotrophic therapy. Nothing close to this ever came, including no myelination and no significant improvement. And so at last, sorry for taking 2 minutes longer than I'm supposed to, many people to thank, but and of course all the collaborators here, but primarily patients advocacy, patients and families that are enabling us to do this work. Thank you. Any questions for me?
Thank you, Paula. This is Bishan. I see one in the chat.
Yes, I see that. Yes, very good point. We're looking at NAA accumulation in brain and oh, yes. So that's a very good question. First of all, with NAA and assessment of NAA, we're still running it and analyzing the data in terms of NAA in the brain and CSF. So data are being generated the next 2 weeks in preparation for the final publication. Yes, so the only, very good question, the only transporter identified in the brain, in the mammalia brain, in any brain, the mammalia brain and human brain and mouse brain is in the astrocytes. And that's the only, so we still don't know how NAA enters oligodendrocytes. And in terms of the effect of high NAA in astrocytes, the result is really the synthesis of NAG. So there is an increase in NAG as well, which is, let's say, I would say toxic or excitatory amino acid that is involved in seizures. Seizure is one of the symptoms in Canavan disease early on. But in terms of pathology, we didn't identify any pathology in astrocytes. So we didn't see any reactive astrocytes in the animal model.
So I had a question, really amazing talk. So actually just 2 questions. One is, do you think that the AAV vector that you used earlier, the neurotropic vector, do you think that the improved, the efficacy was because just more amount was getting into the oligodendrocytes as opposed to previously? And is that why you're getting a better kind of clinical improvement? And the second question is, does the AAV integrate into the genome? So is it just one shot and done, or do you have to just give it a few times so that you have enough dose initially and then stop, or do you expect that you have to give it over the course of the entire, patient's lifetime?
2 very important questions. So in the first clinical trial, we targeted neurons because our hypothesis was the accumulation of NAA would in fact play a role in the demyelination. So we did not, so the first vector that we used in which we saw a great reduction of NAA, but no myelination, targeted only neurons. The promoter was a neuron-specific promoter, so we had no expression in oligodendrocytes. So that's the number one. And so this is when once the animal model was well characterized, we tested the oligotropic vector in the animal model and compare with the neurotropic, and that's when we decided that that was the way to go using the oligotropic therapy. AAV is episomal, does not integrate, so we don't have issues with the potential triggering of oncogenes. And so the vector, the gene is supposed to be the exogenously delivered gene, is supposed to be present and active in the brain until the cell survive. And we have data in production. We had a wonderful donation of a brain of a child that was incredible, compassionate gift of the family of a child that underwent the first gene therapy. And the data is not published, but I can tell you that 22 years after the introduction of the neurotropic vector, there was still exogenously expressed ASPA in the brain. That was 23 years after, because the patient died 23 years after the therapy.
Great. Thank you.
Welcome. Thank you.
I'm wondering about your vector and translating this to other potential diseases. Is anyone using this vector for another target, for another disease? We know that after a couple of different applications, that can be considered a platform, and maybe translation would happen faster for the next. So if we can reach the cell type of interest, what does that path to translation look like for potentially other diseases using this delivery mechanism?
It's a great potential. The vector has been sub-licensed by Pfizer, and so anything that is done with a vector would have to have their approval. We have targets like PND, like ABC, for instance, potentially multiple system atrophy by pretty much reducing or the buildup of alpha-synuclein in white matter. So there are targets, and Myrtle is actually working on developing novel oligotropic vectors. So there is no limitation with the license. But definitely, let's just put aside proprietary data or licensing and everything else. As academic investigator, and that's how I started this with an MTA, and I proceeded to submit an IND as an academic investigator. It was hard. It was not easy. We had meetings with the FDA, interacts, pre-IND, INDs, everything put together. And I worked with 3 consultants. I had an incredible team of investigators and in the lab and outside of the lab. But I was able to do it in 3 years. But it was well designed, and anyone could start a program with this vector without any need for licensing until this become more of a commercial venue when it goes into phase 2, when an IND goes into phase 2.
Excellent. Thank you. That's really helpful. Any other questions from the room? Anything online? All right. Why don't we thank our speaker? Thank you, Paula. Wonderful talk.
Yeah. Thank you so much.